Assalamu alaikum, dear students. Today we will continue with activity 3 of chapter 3, Transcription, First Step of Protein Synthesis, part 2. In this video, we will recognize that the expression of genetic information is done in two steps, transcription and translation. Then we will describe transcription and indicate its location. We will also notice that mRNA forms a non-stable molecule with a transcribed DNA strand. And notice that the mRNA is complement to the template strand of DNA and identical to the non-template strand. Protein synthesis occurs in two steps. One, transcription, followed by translation. Transcription is the step when a copy of DNA, called mRNA, is produced. Keep in mind that DNA remains in the nucleus and mRNA is transported to the cytoplasm. Now let's examine the mechanism of transcription in the upcoming video.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription.
As we saw in the video, transcription includes the separation of the two DNA strands by an enzyme called RNA polymerase, which then assembles complementary nucleotides to one of the two strands, forming mRNA. Finally, mRNA leaves the nucleus and goes to the cytoplasm. As we mentioned before, RNA polymerase is the enzyme responsible for unwinding the two strands, which occurs by breaking down the bonds between them. And as we can see here, it is assembling complementary nucleotides to those found on one of the DNA strands. As we know, the DNA molecule is made up of two strands. One will be used in transcription, and it is called the template or the transcribed strand. However, the other will not be used in transcription, and it is called the non-template strand or non-transcribed strand. After the enzyme unwinds the two strands, it assembles complementary nucleotides to those found on the template strand. The DNA-RNA complex is unstable due to the weak bond between uracil and adenine. And as a result, mRNA detaches from the DNA and becomes free, and now it is able to leave the nucleus. Now, what if I ask you to compare the newly synthesized mRNA to the template or the transcribed strand? What would you say? If your answer is mRNA is complementary to the template strand, then yes, it is right. As you notice, the mRNA has CAU, which are complementary to GTA. UGC, here we have ACG. CAC, here we have GTG. And finally, you can see U, here we have A. So this mRNA is complementary to this template strand. However, if we compare mRNA to the non-template strand, we notice that they are identical for an exception. T in the DNA strand is replaced by U in the mRNA strand. Now let's describe transcription. Transcription takes place in the nucleus by the help of RNA polymerase. In this process, the enzyme unwind the two DNA strands, and then complementary nucleotides are assembled as follows: G2C and A2U. At the end of transcription, many mRNA molecules are produced. The new synthesized mRNA molecule is complementary to the transcribed strand and identical to the non-transcribed one, but instead of T, there is U. Let's have another application. The application says, observe the following document, then answer the questions that follow. The document shows a DNA molecule and a messenger RNA being synthesized. Question 1. Compare the mRNA strand to the coding strand. Question 2. Compare the mRNA to the non-coding strand. Number 3. Match the following terms: coding strand, non-coding strand, to template strand, and non-template strand. You may pause the video and answer the questions. Then we will correct them together. Let's correct them now. If we compare the messenger RNA to the coding strand, we conclude that mRNA is identical to the coding strand of DNA, but instead of T, there is U. If we compare the mRNA to the non-coding strand, we notice that they are complementary to each other. And number 3, if we are to match the terms, we notice that the coding strand is the strand that the mRNA is identical to. And based on previous information, the messenger RNA was identical to the non-template strand. So the coding strand should be matched to the non-template strand. However, the non-coding strand is complementary to the mRNA, which was complementary to the template strand. So the non-coding strand should be matched to the template strand. Let's recall back. The template strand of the DNA is the one that will be used in transcription, then it is the transcribed strand, but it is the non-coding one. And the non-template strand is the non-transcribed one, but it is the coding strand. To sum up, the expression of genetic information is done in 2 steps: transcription and translation. Transcription takes place in the nucleus. Transcription includes the action of an enzyme called RNA polymerase that unwinds the 2 DNA strands, then assembles complementary nucleotides to those found on the non-coding strand. mRNA forms a non-stable molecule with the transcribed DNA strand. The reason behind that is the weak bonds between uracil and adenine. mRNA is complementary to the template strand, which is the non-coding strand of DNA, but identical to the non-template strand, which is the non-transcribed one, that is the coding strand. But for an exception, instead of T, there is U. Thanks for listening.